tackle about our topic about human genetics and genomics. For introduction, the sequencing of the human genome has brought human genetics into a new era of study, resulting in the generation of an explosive amount of information. The human genome, our complete set of genetics information, includes tens of thousands of genes. Some of the very first genes to be identified were those that control of blood type. The objective of our topic is to determine the function of genes and the elements that regulate genes throughout the genome. Find the variation in the DNA sequence among people and determine their significance. Develop and apply genome-based strategies for the early detection, diagnosis, and treatment of disease. Human genetics. Human genetics, this is a branch of biology that studies how human traits are determined and passed down among generations. The study of human heredity occupies a central position in genetics. Much of this interest stems from a basic desire to know who humans are and why they are as they. At a more practical level, an understanding of human heredity is of critical importance in the prediction, diagnosis, and treatment of diseases that have a genetic component, because to determine the genetic basis of human health has given rise to the field of medical genetics in general. Medicine has given focus and purpose to human genetics. So the terms medical genetics and human genetics are often considered synonymous. Chromosomes. Chromosomes are thread-like structures present in the nucleus, which they carries the genetic information from one generation to another. In eukaryotic cells, genetic material is present in the nucleus, in chromosomes, which it made up of highly organized DNA molecules with histone protein supporting its structure. Chromosome structure itself has a pair of each kind of chromosome, known as a homologous chromosome. Chromosomes are made up of chromatin, which they contain a single molecule of DNA and associated proteins. Each chromosome contains hundreds and thousands of genes that can precisely code for several proteins in the cell. The structure of the chromosomes can be best seen during the cell division. Types of chromosomes, they are telocentric, acrocentric, and metacentric and metacentric. So the telocentric, this is the rod-like chromosomes with centromere present on the proximal end, no P-arm or a short arm present. Telometric chromosomes are not found in humans. The second one is the acrocentric. Rod-like centromere present at one and giving rise to one very short arm and exceptionally long arm. The submetacentric, this is the L shape or G shape. So with a centromere near the center of the chromosome, giving rise to two unequal arms. Last would be the metacentric. This is the B shape of chromosomes with a centromere in the middle, giving rise to two equal arms. Function of chromosomes. The main function of chromosomes is to carry the genetic material from one generation to another. Chromosomes play an important role and act as a guiding force the growth, reproduction, repair, and generation process, which is important for their survival. Chromosomes protect the DNA from getting tangled and damaged. So what are the 23 chromosomes? Humans have 23 pairs of chromosomes. 22 pairs of numbered chromosomes called autosomes, and 1 pair of sex chromosomes. That would be the X and Y. So each parent contributes 1 chromosomes to each pair, so that offspring get half of their chromosomes from their mother and half from their father.
What is DNA? DNA, or deoxyribonucleic acid, is a long molecule that contains our unique genetic code, like a receipt book. It holds the instruction for making all the proteins in our bodies. So, deoxyribonucleic acid is a molecule composed of 2 polynucleotide chains that coil around each other to form a double helix carrying genetic instruction for the development, functioning, growth, and reproduction of all known organisms and many viruses. DNA and ribonucleic acid are nucleic acid. And then, the molecule inside cells that contains a genetic information responsible for the development and function of an organism. DNA molecules allows this information to be passed from one generation to the next. DNA is made up of a double-stranded helix, held together by weak hydrogen bonds between purine, pyrimidine, nucleotide-based pairs, which is the adenine paired by thymine and guanine paired by cytosine, also called deoxyribonucleic acid. What is DNA structure? Most DNA is found inside the nucleus of a cell, where it forms the chromosomes. Chromosomes have protein called histone that bind to DNA. So, the structure of DNA. Most DNA is found inside the nucleus of a cell, where it forms the chromosomes. Chromosomes have protein called histones that bind to DNA. DNA has 2 stranded that twist into the shape of a spiral ladder called a helix. DNA is made up of 4 building blocks called nucleotides: adenine, thymine, guanine, and cytosine. The nucleotides attach to each other with A and T and G with C to form chemical bonds called base pairs, which connect the 2 DNA strands. Genes are short pieces of DNA that carry specific genetic information. This is the illustration: types of DNA in the cell. The types of DNA in the cell. There are 2 types of DNA in the cell: autosomal DNA and mitochondrial DNA. Autosomal DNA, also called nuclear DNA, is packaged into 22 paired chromosomes. In each pair of autosome, one was inherited from the mother and one was inherited from the father. There are 4 naturally occurring nitrogenous bases. There are 4 different types of DNA nucleotides: the adenine, thymine, guanine, and cytosine. ECGT is an acronym for the 4 types of bases found in a DNA molecule: the adenine, cytosine, guanine, and thymine. A DNA molecule consists of 2 strands around each other, with each strand held together by bonds between the bases: adenine paired by thymine and cytosine paired with guanine. The sequence of bases in a portion of a DNA molecule, called a gene, carries the instruction needed to assemble a protein. The function of DNA. DNA contains the instruction needed for an organism to develop, survive, and reproduce. To carry out this function, DNA sequence must be converted into messages that can be used to produce proteins, which are the complex molecules that do most of the work in our bodies. So, the function of DNA. DNA is the basis for life on planet Earth. Every living thing has it. The function of DNA is to store all the genetic information that an organism needs to develop, function, and reproduce. Essentially, it is the biological instruction manual found in each of your cells. And then, the function of DNA is tied to its structure, which is helpful to review in order to understand its function. As stated above, the basic building blocks of DNA are nucleotides. These nucleotides are composed of 5 carbon sugar, a phosphate group, and nitrogenous base. The sugars and phosphate link the nucleotides together to form each strand of DNA. When 2 strands of DNA came together, base pairs form between the nucleotides of each strand.
Genetic drift. Genetic drift refers to random fluctuation in the frequencies of alleles from generation to generation due to the chance events. Genetic drift can cause traits to be dominant or disappear from a population. The genetic effect of the genetic drift are most pronounced in a small population. So the genetic drift is when chance events cause changes in frequencies of alleles in a population. The smaller population, the greater the impact genetic drift will have. This is because there are fewer individuals and the gene pool is smaller. If the effects of genetic drift strong enough, the allele might completely remove from the population. So genetic drift when chance events cause changes in frequencies of alleles in a population. So alleles are the genetic variation in a population and the driving force behind the evolution of that population. So this is the example of alleles in this picture. Cause of genetic drift. Genetic drift can be caused by a number of chance phenomena, such as differential number of offspring left by different members of a population so that certain genes increase or decrease in a number over generation, independent of selection. Sudden immigration or immigration of individuals in population changing genes. So the types of genetic drift, the bottleneck effect and the founder effect. So the bottleneck effect is the decrease in the number of individuals in population due to a natural disaster. Genetic drift bottleneck effect usually influences the genetic distribution among the population. Therefore, the effect of genetic drift becomes more significant. So, consequently, the genetic variation among this population will decrease as the number of mating individuals will decrease. Just like what I said, the bottleneck is when a population size becomes very small very quickly. So the founder effect is definition in biology is when a small population may be formed due to the founder effect when a small number of individuals leave their population to start a new colony. These individuals do not necessarily cover the whole genetic set of the population. Therefore, gene drifting effect is significant within this small population. So the population So the population founder effect is when a few individuals in a population colonize a new location that separates from the old population. So this is the types of genetic drift. Difference between bottleneck effect and founder effect. Differences of bottleneck and founder effect. The difference between founder events and population bottleneck is the type of event that causes them. A founder event occurs when a small group of individuals is separated from the rest of population, whereas a bottleneck effect occurs when most the population destroyed. So this is the example of the bottleneck effect in this battle, and the founder effect is small population will destroy. And that's all. Thank you.
Genome. Genome is an organism complete set of the DNA, a chemical compound that contains the genetic instruction needed to develop and direct the activities of every organism. Genome is the entire collection of genes and all other functional and non-functional DNA sequences in an organism in a haploid set of chromosomes. The human genome contains approximately 3 billion of base pairs, which resides in 23 pairs of chromosomes within the nucleus of all our cells. Each chromosome contains hundreds to thousands of genes, which carry the instruction of making proteins. The physical structure of the human genome. Nuclear genome comprises approximately 3,200,000,000 nucleotides of DNA divided into 24 linear molecules, the shortest 55 million nucleotides in length, and the longest 250 million nucleotides, each contained in a different chromosomes. The 24 chromosomes consist of 22 autosomes and the 2 sex chromosomes, the X and Y. Nuclear genome is composed of several linear chromosomes which encodes almost all the proteins required by the cell. Mitochondrial genome is a circular DNA molecule of 16,569 nucleotides, multiple copies of which are located in energy-generating organelles called mitochondria. Mitochondrial is composed of a double-stranded circular DNA molecule which is arranged into a single chromosome. Mitochondrial DNA is encoded for genetic information required by the mitochondria, whereas nuclear DNA is encoded for the genetic information required by the entire cells. Sequencing genome helps the scientists to understand how the genome as a whole and how the genes work together to direct the growth, development, and maintenance of an entire organism. Genome sequencing is figuring out the order of DNA nucleotides or bases in a genome, the order of A's, C's, G's, and T's that makes up an organism DNA. The human genome is made of over 3 billion of these genetic letters. The genome sequencing is referred to sequencing the entire genome of an organism.
What is RNA? RNA is an ribonucleic acid, presents all living cells. Its principal role is to act as a messenger carrying instruction from DNA for controlling the synthesis of proteins. Although in some virus, RNA rather than DNA carries the genetic information. RNA consists of ribonucleotides attached by phosphodiester bands, forming strands of varying length. The nitrogenous bases in RNA are adenine, guanine, cytosine, and uracil, which replace thymine in DNA. Types of RNA. 3 main types of RNA are involved in protein synthesis. Each type of RNA has a different structure that related to its function. First, messenger RNA or mRNA. second transfer RNA or tRNA, plus ribosomal RNA or rRNA, which is essential in protein synthesis. Messenger RNA. So named because it acts as the messenger between DNA and proteins production. When you discuss RNA in school, you're usually learning about mRNA. During transcription, DNA is transcribed into mRNA. This mRNA is then translated into protein. It serves as the genetic template of protein. Since the DNA can't leave the nucleus. Next, we have tRNA transfer RNA. RNA is genetically single-stranded, but tRNA actually folds back into itself to form a distinct T-like shape, without going into two-deep transfer. RNA is responsible for bringing amino acid together during translation to form the growing peptide chains that will become a protein. And last but not least, we have rRNA or ribosomal RNA. This RNA is the main component of ribosomes. RNA combines with special proteins to form ribosomes, which then read mRNA to form proteins. But essentially, ribosomes contain rRNA and help build proteins. To bring it all together, sections of DNA are transcribed into mRNA, which is brought from the nucleus to the ribosomes, composed of rRNA and proteins. The mRNA is then read by the ribosomes, which brings the tRNA in with the appropriate amino acid to build whatever proteins the mRNA is template for. Function of RNA. RNA transfer genetic information from nucleus to the cell, makes ribosomes, help assemble proteins. RNA carries a broad range of functions, from translating genetic information into the molecular machine and structures of the cells to regulating the activity of genes during development, cellular differentiation, and changing environment. RNA is of unique polymers.
So, if we say heredity, it is a way that one person can pass his or her characteristic from one to the next generation by their genes. Genes is a way that process all the information, specific making of proteins that are responsible for the traits of individual. For example, is if you are a parent, the attitude or the way you as a person, the next generation of yours can get some of your characteristics like the color of your hair and eyes, the shape of your nose and ears, as well as your body type and size.
There are two types of heredity. First is simple heredity. In this type of heredity, ang isang gene, it controls one trait of alleles that either dominant or recessive. If we say dominant, it is the trait that appears in the offspring. Offspring only needs one dominant allele from the parent to show up, while recessive are traits in the background or may not show up in the offspring. Only show up in the offspring gets two recessive alleles from their parents. Example are height and color in pea plants. Next is second is complex heredity that included polygenic inheritance, condominance. This polygenic inheritance, this is a slow process requiring many generations to achieve desired result. Example is height of humans. Next is condominance. In this type of complex heredity, it needs multiple of alleles for given that trait, but these are not expressed to one another but in combination. Example is RR or red flower and
WW or white flower equals RW or red and white strip flower. Incomplete dominance. This is similar to condominance but except in the characteristics of alleles blend instead of remaining distinct. Example is RR or red flower and WW or white flower equals RW or pink flower. Process of simple heredity. Heredity also is best described as inheritance that can pass from the parents to their offspring. Through direct outcome of heredity that are random genetic, the recombination of sexual reproduction that can only function during sexual reproduction and ensures genetic diversity. Although heredity and environmental influences determine of the animal but it influences the overall quality of an animal. Heredity in agri-science. In agri-science, in Hodge factor, they are successful in agriculture in selective breeding programs in processing to create high quality of hybrid offspring. Their plants and animals inbreed through several generations to isolate specific trait or traits and do more than 7 generations to prevent genetic disorders. In final generation of 2 lines inbreed for different traits are crossed in producing offspring with the beneficial traits in both lines. These are the result of offspring possesses hybrid vigor. If we say hybrid vigor, it is last only 1 generation as hybrid organism rarely express through or 2 type of offspring. Alternative forms of the gene resurface in the new cross. That's all, thank you.
So in this video, I will discuss about the nucleotide, its structures, examples, and of course its function. So first, what is nucleotide? A nucleotide is an organic molecule that is the building block of DNA and RNA. They also have function related to cell signaling, metabolism, and enzyme reaction. A nucleotide is made up of 3 parts: a phosphate group, carbon sugar, and a nitrogenous base. The 4 nitrogenous bases in DNA are adenine, cytosine, guanine, and thymine. RNA contains uracil instead of thymine. A nucleotide within a chain makes up the genetic material of all known living things. They also serve a number of functions outside of genetic information storage as messengers and energy moving molecules. The next one is nucleotide structure. Nucleotide structure is simple, but the structure they can form together is complex. An image above is an image of DNA. This molecule consists of 2 strands which wrap around each other, forming hydrogen bonds in the middle of the structure for support. Each nucleotide within has a specific structure which enables these to form. So let's proceed to the next one. As what I've said earlier, nucleotide is made up of 3 parts: nitrogenous base, carbon sugar, and a phosphate group. But first, let's talk about the nitrogenous base. The nitrogenous base is the central information carrying parts of the nucleotide structure. These molecules, which have different exposed functional groups, have differing abilities to interact with each other. As in the image earlier, the idea arrangement is the maximum amount of hydrogen bonds between nucleotides involved. Because of the structure of the nucleotide, only a certain nucleotide can interact with other. The image earlier shows thymine bonding to adenine. The image above shows thymine bonding to adenine and guanine binding to cytosine. This is the proper and typical arrangement. This even formation causes a twist in the structure and is smooth if there are no errors. One of the ways proteins are able to repair damaged DNA is that they can bind to uneven spots within the structure. Uneven spots are created when hydrogen bonding does not occur between the opposing nucleotides molecules. The protein will cut one nucleotide and replace it with another. The duplicate nature of the genetic strands ensures that the errors like this can be corrected within a high degree of accuracy. The second portion of the nucleotide is the sugar. Regardless of the nucleotide, the sugar is always the same. The difference is between DNA and RNA. In DNA, the carbon sugar is deoxyribose, while in RNA, the carbon sugar is ribose. This gives genetic molecules their name. The full name of DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. The sugar, when it's exposed to oxygen, can bond with a phosphate group of the next molecule. They then form a bond which becomes the sugar phosphate backbone. These structures add rigidity to the structure. As the covalent bonds they form are much stronger than the hydrogen bonds between the two strands. When the proteins comes process and transpose the DNA, they do so by...
Good day. In this video, I will describe about genetic diversity. So, genetic diversity, each species is composed of individuals with their own set of genes. A gene is the inheritance unit by which generations can inherit the features of their parents. It refers to the nucleotide sequence in the DNA strands packaged in chromosomes. So next is genetic variation. So what is genetic variation? Genetic variation is defined as the presence of a various number of alleles with different distribution frequencies in a certain population. Genetic variation simply refers to the variation of genomes at the individual level. So next is genetic diversity and biodiversity. So what is the impact of genetic diversity on biodiversity? So genetic diversity is the main contributor to biodiversity in the ecosystem. The term of biodiversity, it refers to the biological diversity of life on Earth, and therefore that includes multifarious microorganisms, plants, and animals, along with their genes and their respective environments or habitats. And many researchers define that the genetic diversity as the amount and extent of genetic variability among populations, whereas biodiversity refers to the diversity of all organisms in a given. And the next is the importance of genetic diversity. So, why is genetic diversity is important? Genetic diversity is important because it could ensure that certain groups of individuals, species, or population will be able to adapt to certain environmental factors. Besides having to deal with environmental conditions, genetic diversity renders them the ability to resist emerging disease and epidemics. On the contrary, low genetic diversity may increase the susceptibility of a species to biotic and abiotic stresses such as disease and drought, and therefore facing a higher risk of extinction in the long run. And next is the the example of genetic diversity. So, genetic diversity is significant in humans in essence, or humans have varying eye colors, hair color, skin color, height, or etc. Diversity in human population is the key to identification techniques such as using fingerprints. And the last is the factors of genetic diversity. Genetic diversity is influenced by four events controlling evolution: mutations, genetic drift, gene flow, and natural selections. However, only mutations can produce completely new alleles.
Hi, good day. The topic that I am going to present to you today is all about genomics. What is genomics? Genomics is an area within genetics that concerns the sequencing and analysis of an organism's genome. It involves the study of all genes at the DNA, mRNA, and proteome level as well as the cellular or tissue level. The term genomics was first coined in 1986 by Tom Roderick, a geneticist at the Jackson Laboratory in Maine, during the meeting about mapping of the human genome. Genomics is the study of all genes present in an organism. By definition, it can be defined as a discipline in genetics that applies recombinant DNA sequencing methods, and bioinformatics to sequence, assemble, and analyze the structure and functions of genome. It includes the studies of intragenomic phenomena such as heterosis, epistasis, pleiotropy, and other interactions between loci and alleles within the genomes. Okay, let's move on to the history of genomics. Genomics is a concept that was first developed by Fred Sanger in early 1970s, who first sequenced the complete genome of a virus and of a mitochondrion. In 1972, Walter Fires and his research group became the first to sequence a gene. They sequenced the gene of bacteriophage MS2. And in 1995, Hamilton O. Smith and his team became the first to sequence a genome of a free-living organism, that of Haemophilus influenzae. Okay. There are 4 types of genomics in my research. These are the structural genomics, functional genomics, comparative genomics, and mutation genomics. Okay, structural genomics is a field of genomics that involves the characterization of genome structure. The aims of this structural genomics is to determine structure of every protein encoded by the genome. Functional genomics aims to understand the complex relationship between genotype and phenotype on a global genome-wide scale. And comparative genomics is a field of biological research which the genomic features of different organisms are compared. Mutation genomics is the study of genome in terms of mutation that occur in an individual DNA or genome. Okay, now let's move on to the goals. The main goal of genomics is to sequence the entire genome by cutting it into small, manageable pieces or fragments. Next goal is assemble the entire genome from the pieces. And the other one is understand how gene expression takes place. Why to sequence genomes? Okay, sequencing genome helps understand how the genome as a whole
Types of genomics. Structural genomics is a field of genomics that involves the characterization of genome structures. Structural genomics can be used to predict novel protein folds based on other structural data. And also, it takes modeling-based approach that relies on homology between a known protein and solid protein structure. Functional genomics aims to understand the complex relationship between genotype and phenotype on a global genome-wide scale. Functional genomics is the study of how genes and intergenic regions of the genome contribute to the different biological process. Comparative genomics is a field of biological research which genomic features of different organisms are compared. Comparative genomics is a field of biological research in which researchers use a variety of tools to compare the complete genome sequence of different species. Mutation genomics, the study of genome in terms of mutation that occur in individuals' DNA or genome. A mutation is a change in DNA sequence that result from DNA copying mistakes made during cell division, exposure to ionizing radiation, exposure to chemicals called mutagens, or infection by viruses.